Hey everyone, and welcome to this explainer. Today we're diving into something truly wild. We're unpacking the messy, intensely fascinating biological conflict that sits right at the very heart of cloning. We'll be looking at decades of research, starting all the way back with the earliest amphibian experiments, moving through the global shockwave that was Dolly the sheep, and landing right at modern human stem cell derivation. The absolute core of all this research revolves around a technique called Somatic Cell Nuclear Transfer, or SCNT. Now, this is a technology that totally flipped our foundational assumptions about how cells work on their head. But, as you're going to see, it comes with a pretty massive biological catch.
So let's just jump right in by asking the big, core philosophical question that our source material presents right out of the gate. For the longest time, biologists were totally stuck on this puzzle. Basically, when an embryonic cell differentiates and becomes, say, a skin cell or a heart cell, does it permanently lose the rest of the genetic instructions it needs to make a whole organism? Or are those instructions just kind of locked away somewhere?
Well, the mind-blowing answer, the one that fundamentally reshaped developmental biology forever, is that the DNA is fully retained. The whole blueprint is still there. Differentiation is not this irreversible loss of genes. But here's the shocking truth. Trying to force that specialized adult cell back into a blank embryonic state is incredibly prone to failure. Our sources point out that even though the genetic blueprint is completely intact, the success rates for healthy live births in cloned animals absolutely plummet to a tiny, tiny minority. It's famously almost stubbornly inefficient. So, we have to ask, why? That brings us right to section 1, the cloning paradox. To really understand why cloning fails so often, we first need to look at the actual mechanics of the procedure. It's all about artificially forcing two completely different biological states to collide. Somatic cell nuclear transfer relies on these four extremely delicate steps. First, you isolate a specialized donor cell, like that skin cell we talked about. Next, you carefully remove the nucleus from a recipient egg cell, essentially vacuuming out its maternal chromosomes. Third, you drop the donor's nucleus right into that empty egg. And finally, you have to artificially activate this newly reconstructed embryo with a chemical or electrical spark, just to trick it into thinking it's been fertilized. And honestly, every single step in this process is a huge biological shock to the system.
And that beautifully sets up section 2, the battle for supremacy. Because as it turns out, this transfer isn't just some simple, peaceful swap of genetic puzzle pieces. It literally kicks off an epic microscopic conflict inside the cell. Nobel laureate John B. Gurdon, who was an absolute pioneer in those early cloning experiments, actually framed this biological process as a war. He famously called it a battle for supremacy between two opposing cellular forces. The egg isn't just passively sitting there taking orders. This is an all-out fight for the very identity of the cell. You've got this incredible biological mismatch happening. On one side, there's the stubborn somatic nucleus. It carries a whole lifetime of developmental history, and it is desperately fighting to retain its specialized identity. And then, on the exact opposite side, you have the recipient egg, acting like this aggressive reprogramming machine. It is absolutely loaded with maternal factors that are trying to violently strip away all that history and force the nucleus back to a blank slate, usually in just a matter of hours. Let's move into our third section to see how the cell fights back: epigenetic memory and resistance. So how exactly does an adult cell defend its identity? It uses something called DNA methylation. Basically, these are chemical tags added directly onto the DNA that act like padlocks on certain genes. Think of this transcriptional inertia like a really stubborn tenant who absolutely refuses to hand over the keys to their apartment. In a cloned embryo, if the egg doesn't completely erase these somatic tags, the crucial developmental genes the new embryo desperately needs to grow just stay locked. The doors won't open. But it actually goes even deeper than that. The cell also relies on histones, specifically these deeply repressive marks like H3K9ME3. You can think of these as proteins that spool the DNA up into this dense, inaccessible physical armor. Our source material makes it clear that these histone marks actively and strongly resist the egg's rapid attempts to reopen those embryonic genes. We are talking about physical barriers that just flat out refuse to be remodeled. The genetic information is technically all right there, but it's buried under layers of molecular concrete.
Meaning, the real challenge of cloning isn't just moving DNA around with a tiny needle. Not at all. It's forcing that donor nucleus to experience total and complete cellular amnesia. The egg has to aggressively scrub away the DNA methylation and tear down that histone armor before the embryo starts dividing, and the clock is ticking. If the cell remembers even a little fraction of its past life, say as a memory cell, the entire new developmental program just derails completely.
Which brings us to the ultimate test in Section 4, the extra embryonic gauntlet. Because look, even if the egg seems to win that initial battle and forces some level of amnesia, the cloned embryo still has to survive the absolutely brutal developmental checkpoints of gestation. You can see the attrition rate right here on this chart, and it's terrifying. Early success in cell division, a phase called cleavage, is actually relatively high because the egg's maternal proteins carry a lot of the load. It might even make it to the blastocyst stage at a decent rate. But the second the embryo hits the milestones of implantation and ultimately live birth, man, the success rate just completely falls off a cliff. Most cloned embryos fail this gauntlet, specifically because tissues like the placenta demand unbelievably precise epigenetic regulation. A partially amnesiac cell just cannot provide that level of tightly timed control. And that leads to recurrent, devastating failures across all sorts of different species. We're talking massive placental enlargement, faulty imprinting, large offspring syndrome, and neonatal respiratory failure. The embryo might look totally fine early on, but those hidden reprogramming defects inevitably rear their ugly heads when the complex demands of fetal survival kick in. So let's wrap this up with Section 5, the future of reprogramming. What does this steep drop-off mean for the actual value of this technology? If cloning is this punishingly hard, is it all just a biological parlor trick? No way. Absolutely not. But we have to separate our end goals. Reproductive cloning, making a whole new animal, requires full extra embryonic development and term gestation, which gives us that insanely high failure rate. But therapeutic cloning, that changes the whole game. It only requires early blastocyst formation to harvest patient-matched embryonic stem cells. The thesis highlights a huge point here. For therapeutic cloning, a partial victory where the cell forgets just enough of its past to become a stem cell, but maybe not quite enough to form a placenta, is totally 100% sufficient. So even with all its inefficiencies, SCNT remains incredibly powerful and scientifically indispensable. It's the ultimate benchmark for complete reprogramming. It exposes the rules of cellular plasticity, showing us exactly which genomic regions are easy to reset and which ones are stubbornly locked down. It actually informs our modern engineered stem cell methods by showing us the upper limit of what nature can pull off. Plus, it lets us build highly valuable disease models. SCNT might be a really tough way to consistently clone livestock, but it is without a doubt the gold standard for teaching us how cellular identity is stabilized and how it can be reversed. So as we finish up this explainer, we're left with this really profound reality. We know how to clone. The mechanics work. But the ultimate barrier isn't missing DNA at all. It's a specialized cell's absolute, armor-plated refusal to forget who it was. So the big question is, will we ever fully master biological memory? Because the secret to the future of biotechnology isn't just about writing new genetic instructions. It's about figuring out how to successfully erase the stubborn memory of the past. Thanks for exploring this complex topic with me, and keep questioning how our biological world works.